All right, guys, let's talk about review stuff. That's a little loud. Okay, first things first, not related to review at all, but Dr. Corinne Gill heads up an organization here at Baylor that just got started this semester, and so I was going to promote this to you guys. Obviously, it's after the April 6th, 26th interest meeting, but if you guys still want to, what it is, it's a Baylor chapter of the Society for Advancement of Chicano, Hispanic, Latinos, Native Americans in STEM. So it's kind of a very interesting group that they're trying to basically promote more involvement in STEM and bring more people into STEM, especially for high school and undergraduate, as well as graduate school. And so it's actually a really interesting club, and Dr. Gill is very, very good at explaining things and has kind of a wealth of knowledge in this society. But I was going to, make this known to you guys so that you guys have an opportunity to go and maybe check it out or apply and see what it is. One of the things that she wanted to stress is it's not just for people who are, Hispanic or Chicano or Native American. Anybody is welcome to come. The main goal is to help provide, fundraisers and events to help people bring more people into STEM from communities that is supporting. So it's not just for that. So if you aren't part of, somebody who's Hispanic or Native American, you can still come and participate and help increase that. So there's the email right here for this for interest, as well as you could also just message Dr. Corinne Gill. She's very, very nice, and she won't mind being bugged at all about this because this is her little project to get up and running. So they just got started this semester, and so they're trying to get more people in. So if you guys are interested in something like that, definitely try checking it out. Okay, that being said, one of the things I was going to go over, I didn't know if you guys were particularly interested in, I know some of your TAs kind of went over some of this. Did you guys have any questions about this thing, and you wanted me to kind of go over and fill in some blanks on here, or if you guys were like pretty well set up with the practical? This is the thing that's on the review that's on Canvas for you guys right now. So did you guys need help going through that? Is there a bunch of blanks? Okay. Do you just want me to kind of roll through it, the entire thing? Okay, I'll do that. Now, before I do that, real quickly, I'm going to talk about the basic format of the exam. It's going to be very similar to the first lab practical. We'll have stations. Each of those stations will have between 1 and 4 questions. There will be a bonus station, which will have stuff made up from the Cool Science Thing of the Week. With this, there is kind of, each of these things will be based off of something that we learned this semester. It's not going to be like random stuff from the beginning of the semester, but it will be cumulative in the sense that we had to use MSA plates this half of the semester to do identifications, right? So you should be able to interpret an MSA plate, know what it selects for, know what it differentiates for. You should know how to look at a gram stain and go, oh yeah, this is gram positive, this is gram negative, this is a cosi, this is bacillus, this is the arrangement. But I'm not going to be like, hey, who invented the gram stain? Christian Graham, 1888. Anyways, okay, see, you don't need to know that. So we'll go over a couple of these, and then, excuse me, I'll maybe go over a couple questions that you might actually see in the exam. Not exactly in the exam, but kind of the style of them. So the first one up, rapid tests, which we wanted to do right at the beginning of the semester, and people forget all the time. I know, because it's not very common. These lights not work? Nope. But there's lights for this specific thing. Oh well. Anyways, oh yes, that should be. Yeah, there we go. Okay, so if I have to draw anything. Okay. Rapid tests are what? What do we call this? And a lot of people kind of struggle with this, I know, from the 4102. Do you guys know what this is called? Immunosorbent. So they're all immunosorbent. So the entire words that we're looking for, if you guys remember, they're ELISA tests, which is an acronym for enzyme-linked immunosorbent assay. And we just kind of abbreviated it to immunosorbent assay, that would be fine. But you should know this abbreviation. So this is a good one to know. Enzyme-linked immunosorbent assay. Okay. the rapid test for blank and the rapid stress tests are for blank, and it looks for blank antigens. I know, that was really confusing. Blank with a dye attached to it, and then blank is embedded in the test strip. So we know it's a test strip. Which immunotest did we use that had a test strip? What was the test strip one for? Which bacteria? Okay, it could either be strep or staph. Who says strep? Who says staph? Who didn't vote? I'm going to pick on you guys if you don't vote. I did in the last class. So it is strep. So strep is the one that is the strip test. So strip test, we take a little swab it around, in this case your throat, and then we put in some liquid, and then we take the test strip and dunk it in the liquid, and we wait for it to suck up the liquids. So while we do that, make sure I'm actually recording the correct thing. Yes, I am. Good job. All right. So while it's doing that, we're looking for a certain type of antigens. And these are antigens specific for Streptococcus. What are those antigens called? Somebody know? Oh. Group A, yes. The abbreviation is GAS. Group A strep antigens, so GAS antigens. Very good. Okay, so we're looking for those GAS antigens. We put in here, in this liquid, when we swirl the swab around in there, there's a liquid, and we put in it a antibody, and the antibody has a small label in it. So that's what we put on there. That's the labeling part. So we put in an antibody that's got a little dye on it, and that little dye shows up a certain color. In this case, I think it's for your guys' test strips, they were blue, but sometimes it could be red or whatever color we want, honestly. And we put in there, and this antibody is specific to GAS antigen, so it will only bind GAS antigens. So if you have something else that's not a GAS antigen, this guy's not going to stick to it. And then there is a test section and a control section. The control section is always after the test section. So control is always over here, test is always over here. And this is actually really, really important, because if you have the control thing in front of the test, we would never know if the antibodies got all the way to the test region. So the reason why we have it in this order is because when we add this into the liquid, it's going to come up from this end and it's going to go this direction. We have to have the control region over here, because the control region tells us, hey, this antibody, even if it's not bound to anything, this antibody that's dyed, it made it all the way up here. But if we had the control region in front of the test region, we would never know if this antibody ever made it to the test region. We'd have no idea. So that would make it, that's why it has to be after the test region. So we know that the liquid's gone all the way through past the test region and into our control region. So that's why we have it after. Now the control region and the test region have two different things embedded into them. For the test region, what do we have embedded on the test region? Does anybody remember? I'll give you guys a hint. It's very similar to the labeled antibody. It is, in fact, exactly like the labeled antibody, except it just doesn't have a label. So it is anti-gas, and it binds gas antigens. So if gas antigens are present, that guy will stick to it and hold the gas antigen in place, and eventually our dyed antibodies will bind to that, and it'll create that little line if it's present. For the control region, what do we have in the control region that's bound there? I'll give you guys a hint. It's very similar to these two, but different in only one way. It's also an antibody. So it's also an antibody, but this antibody is special because this antibody binds other antibodies. So it will bind this guy all by himself. It'll stick him on there. And that just makes sure that our dyed antibody made it all the way through here. This guy is just making sure that the gas antigens are bound, and then the gas antigens will also be bound by our dyed antibody. So the test region has antibodies that are against gas to trap both gas and the gas that's bound to our dyed antibody. The control region has antibodies that just bind our antibody that's labeled. So that's what it does. So it's a little confusing, I know, but that's the setup. Does that make sense? Kind of, maybe a little. There is an animation on Lab Archives that does tend to help with this, clarify it, because I make animations way better than I make drawings by hand, but that's what's going on. Does anybody have any questions on how that's working? Does that make sense, more or less? Maybe? You guys seem very quiet. You guys can tell me if I don't make sense. It's fine. I won't take it personally. Very personally, yeah, a little personal. No? Okay. All right. We'll test your knowledge in just a second. Okay, so rapid staph tests looks for something. Yeah, so it's actually looking for it looks for Staphylococcus aureus specifically. So that's what it's actually looking for. The test uses some kind of bead. It's a special kind of component of the bead. These beads are made of what? Latex, very good. These beads have certain chemicals embedded on them. Does anybody remember what those chemicals are? They have antibodies bound on them, and they also have fibrin on them. So these test ones are kind of weird because they're like, so they're just little beads, and on the beads they have these antibodies stuck on them, and they have these little tiny bits of fibrin on there. Okay, now what makes these unusual is the antibody is not binding anything. Antibody is not doing the binding. The Staph aureus has the ability to bind antibodies. It's got this wacky thing called protein A. Protein A binds onto antibodies because it kind of helps protect itself by hiding, like it's coating itself in like pretend armor to say, look, I'm actually part of your body, don't attack me. So it's kind of hiding in plain sight by coating itself in antibodies. So we put antibodies on here, and the Staph aureus can bind these antibodies, which is really handy for doing this test. So Staph aureus will have its own little thing here, and it's got this little tiny protein that comes out, and it likes to bind antibodies. It also has this ability to break down fibrin with an enzyme, and this little enzyme likes to chew this guy up and turn it into fibrinogen, which makes this kind of like mesh. And so what ends up happening is these beads get all interlaced with each other and make a big kind of agglutinated web. So it's got all these kinds of little dots that are really easy to see. If Staph aureus isn't in the presence, then you won't see those dots. It'll just kind of be an evenly colored, kind of smooth liquid. It won't have any dots in it, so it won't have any agglutination. So that's how we do this test. Does anybody have any questions on those things? Did I skip any blanks that you guys need clarified? Yes, no, maybe so? Okay. Let's talk about unknown isolation. So you guys should know the general process that we do for unknown isolation. The very first thing we do, you are given 3 plates to do your initial isolation, because you don't know what it is. You don't know if it's gram-negative or gram-positive. So we give you 3 different plates. On those plates, you're supposed to do a specific type of streak pattern. What is that streak pattern called? Streak for isolation. Very good. All right, good. You guys are on top of this. Streak for isolation. And you guys should remember that pattern. It's really easy to remember. That pattern is just a circle and a line across and a line like this. And then it goes up and down like this, so that we're going from high concentration to low concentration. And then you go across a couple lines going from high concentration to low concentration. And then the same thing over here, high concentration to low concentration. So eventually get an isolated organism. Now we give you guys 3 different plates because we don't know what your organism is, either gram-positive or gram-negative. So we're going to give you like kind of a broad kind of shotgun approach. So we're going to give you 3 types of plates. One's just nutritive. It just gives you nutrients and lets bacteria grow on it no matter what. What is that plate called? TSA, yes. It's the same plate that we use for pretty much everything in lab. Anytime we give you a slant or a plate, it's almost always TSA. Now, aside from this, we give you 2 different types of plates that have that only allow certain organisms to grow on them. The first one allows gram-positives only to grow on it, and that plate is called a I'll give you a hint. It's 3 letters. PEA plate, yes. So phenyl ethyl alcohol. So remember that. That's an important one. So it only allows gram positives to grow in it. Kind of. It doesn't do a great job, but it does a pretty okay job. So it only allows gram positives. And this is a selective only plate. It doesn't grow anything else on there. It doesn't do selection on there. It doesn't do anything like that. It's just, hey, I only allow this to grow on there. The other one is for gram negatives, and that plate is called McConkey's. Okay, so you guys should all be like fighting to answer this because this is things that you should know off the top of your head. So if you are struggling with this, if you're like, I don't know what gram negative plates, the selective plate for that is, you should be like, oh, I really need to study this and focus on these ones. So it should be a gram negative. It's McConkey's auger plate. Now, McConkey's auger is selective, but is it also differential? What do you guys think? Who says it's differential? Who says it's not differential? Who didn't vote? Okay, stop not voting. We're gonna do it again. So I'm gonna call on you for answers. Okay, so is McConkey's auger differential? Who says it's differential? Who says it's not differential? Okay, it is differential. You guys are right. So it's a differential plate. What does it differentiate? Lactose what? Fermentation. Good. So it can be a strong fermenter, it can be a weak fermenter, or it can be non-fermentative. Now, after we do this, we can take isolated colonies, we can gram stain them, except for on the McConkey's plate. And why can't we do it off McConkey's plates? Yeah, because it has a dye in it. So it's got dyes built into it, and that dye will make the gram staining not very clear. So we don't want to use that one if we can avoid it. So how do we differentiate on like a TSA? Because TSA isn't a differential media. You just kind of have to look at the colony characteristics and go, oh, this one looks different than this one, so this one's probably gram negative, this one's gram positive, the other one, what have you. And for you guys, because you guys didn't have mixed cultures, just whatever grew on whichever plate, you went with that one. Okay. Okay, gram stain. You should be able to look at gram stain and go, I know what these are. So our first plate, we're gonna do another voting thing. First one here, is that gram negative or gram positive? Who says gram negative? Who says gram positive? Who didn't vote? You two didn't vote. All right, it is gram negative. Now, for those you two who didn't vote, and I saw you didn't vote, what is the shape of this organism? Yep, the red one. It's actually cosi. So I know it's really hard to see on that one. I'd give you guys a little bit clearer image. You can see it doesn't have to squish, it's got the little circles on here. But I'll give you guys another chance because there's that follow-up question. What is the arrangement for this guy? So what do you guys think the arrangement is? It could be Staphylo, it could be Strepto, it could be Tetrad, it could be random, Palisade. No, it is not Staph. It is Strepto. So Strepto is the lines, the change. So Streptococcus is those chains, long, nice chains. So we call those Strepto. The middle guy, I'm going to enhance this just to make it easier. All right. Is this gram positive or gram negative? Who says gram positive? Who says gram negative? Who didn't vote? Okay, nobody put their hand up that time. All right, I'm going to make it harder. Yes, it is gram positive. And this one is gram positive cosi as well. But this one has a different arrangement. And what is this arrangement called? Staphylo. Very good. All right, so Staphylo is the arrangement here because it's little clusters. You can see you get these repeating patterns of clusters. Look like kind of clusters of grapes. If you grab grapes off a vine, it would kind of look like this. Okay, very good. Last one. This one's kind of weird. Wacky. Hopefully you guys know by now, this one is in fact gram positive. That one's easy. And you can see that they're circles. But it's got a unique type of arrangement. And we only see it in 2 of our organisms that we did for our unknown organisms. What is this arrangement called? Tetrad. Very good. People in the front row, all of them said it. Tetrad. Do you guys know which 2 organisms showed up as tetrads in our unknowns? That one's kind of a hard question. There's only 2 organisms that are tetrads. It's Micrococcus luteus and Cocuria rosea, or Micrococcus roseus, depending. It's the same organism, different names. But those are the only two that show up as tetrads. It's kind of an odd one. And they actually asked me this in the last class, and I thought I'd point this out here real quick. The reason why we know that this is tetrad and not something like random or diplo, because you can see like a couple of pairs here, we look at what the average is. So by chance, if you see just like one group of 4, that's just nature, it's just random, not likely to happen. But if you see multiple groups of 4, so this guy, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, that's not random chance, that is a pattern. And so we would definitely say that this is a tetrad. It's not just random chance that we're getting patterns of 4 over and over and over again. That is a consistent arrangement. Okay? All right. Does anybody have any questions on those? Makes sense. Yes? Sorry, what was that? You don't have to know them, but if you were like, hey, Josh put an unknown question on here and one of them happened to be gram-positive tetrads, that would cut down your choices considerably, right? There's only there's only 2 organisms that you have to look at now. And I'll give you guys even a greater hint. One of those organisms is yellow and the other one is pink. Thus the name roseus, rose-colored, pink. So if you guys saw a plate that had tetrads and you were like, hey, that's pink, that has to be roseus. Kind of cuts down on your workload. So, not necessary, you can do it through other tests, but that is kind of like the easy way to do it. Okay, and I always like doing things easier if I can. Okay, gram-positive test. The enzyme blank is responsible for differentiating the 2 major groups of bacteria in the lab. What enzyme is it that we're looking for? What's the major differentiation for gram-positives? What is it? Catalase, very good. Catalase is a very important enzyme, and it helps you process which chemical? Let me pick on you again. It's catalase, so what does it process? What does it break apart? You don't remember? So it actually breaks apart hydrogen peroxide, right? Which is a naturally forming radical. It kills bacteria, right? We use hydrogen peroxide in wounds. If you're really tough, you'll put it straight into your wound and it'll bubble up. But if you are an organism that doesn't deal a lot with oxygen, you actually tend to not have to use catalase. So you tend to be catalase negative, because it's an enzyme you don't need to work with if you prefer things to be without oxygen. And what type of organisms generally are speaking, they don't like oxygen? What do we classify them as? Anaerobes, yeah. In this case, the ones that we have, you could be like strict anaerobes, or you could be like facultative anaerobes. Most of ours are facultative anaerobes that don't like a lot of oxygen, and they can kind of survive, but they like grow really weakly. So if you guys had organisms like Streptococcus pyogenes, or what's another one? Streptococcus agalacti or Enterococcus faecalis. Those don't like a lot of oxygen, and they grow really weakly when we put them in oxygen because they're like, oh, we have to work so hard to stay alive because we just don't have the enzymes to do oxygen very well. So they tend to grow more weakly. But if you look at the ones that are catalase positive, Staphylococcus aureus, Staph epi, Micrococcus luteus, Cocardia rosea, those grow really robustly and very thick and nice colonies because they have the ability to handle that oxygen better. Okay. So each of those, if you're catalase positive, you have one set of tests. If you're catalase negative, you have a different set of tests. But actually, if you're catalase positive or catalase negative, you guys share one test between both of them. They get the same test. That test is for what? What do we call that test? It's the only broth test? PYR, pyre. Okay. So the pyre test is really, really handy because we're looking for the ability to break down pyre reagent. So with an enzyme called pyridonyl aminoglycosase, which is like really, really long. You guys don't have to worry about that. It's just pyre enzyme. We're looking for the enzyme pyre. So we're looking for the ability to break down this, and the end product we add with a reagent, which we call pyre reagent. If you really want to know the long name, it's like dibenzoamino cinnamaldehyde, which is incredibly long. It's orange in color normally, but when it reacts with the pyre reagent that's been cleaved, it creates a nice bright red color. And that bright red color means it's positive. That bacteria has the enzymes so it can cleave it, and that cleavage allows the reaction to happen to make that bright red color. So red means positive, orangey yellow just means negative, nothing happened. Okay, makes sense? All right. other than this, the catalase positives have a certain set of tests and the catalase negative have a different set of tests. The big one to know is the antibiotics that each one has. So the catalase negative, they like, we use a specific type of antibiotic for their test. Does anybody remember what the antibiotic is for the catalase negatives? I'll give you a hint. It's one of the most commonly prescribed antibiotics, especially if you get like an open wound or something. It's the first one that they go for. Bactrim, yes. SXT is the other word for it. Sulfasoxazole trimethoprim. This is what SXT stands for. Yeah, Bactrim. So that's for the gram-positive catalase negatives, but for the catalase positives, we have a different set of antibiotics that we test for. So what's the other antibiotic that we use? What is that? No? It's got a different name to it. Novobiacin is correct. Very good. So novobiacin is the other one. So we test novobiacin on a regular old Mueller-Hinton auger plate, which is the normal plate that we do for the antibiotics. But for the catalase negatives, we do them on a special plate. That special plate is, what is that special plate called? It's bright red. Blood auger, very good. And so that's like a 2-for-1 test because it also gives us information about your ability to lyse red blood cells. So it tells you if you're hemolytic or not. What are the 3 options for hemolysis? Alpha, beta, and gamma, or negative. Negative's fine. Non-hemolytic. So if you're non-hemolytic, you do nothing, you just grow on there, nothing changes. If you're alpha, you kind of create this kind of brownish color where you're starting to degrade the red blood cells, but not very much. Beta is completely see-through. So if you hold your hand behind it, you can see your hand through the auger. It's completely clear. And it's because it's lysed all the red blood cells. Very good. Okay. Other thing in here, the slant. We use a slant. It's called BEA. What does BEA stand for? Ooh, this is a hard one. Okay. BEA stands for bile esculin auger. And we're looking for the ability to break down which chemical? Esculin, very good. Breaking down esculin and esculatin. And if it does break it down, then it reacts with the media's components to create a nice dark brown coloration. If that dark coloration is present, that means that you are positive for esculin. If it doesn't change color, then it's negative. Excuse me, it's the easiest test, super straightforward. MSA plate is one of the other ones that we use. MSA plate, you guys remember, if it grows on there, it's gram-positive halophile. If it doesn't grow on there, it's not a halophile. If it does grow on there and it changes from red to yellow on the media, that means that it ferments a sugar. Which sugar does it ferment? Mannitol, very good. So that's important to know. So if it's mannitol positive, it'll change that nice bright yellow media. If it's negative for mannitol fermentation, then it just stays red. The last one, and I know a lot of people had trouble with this, is the DNase one. And I know DNase is a little confusing. So DNase auger, it is unique because it has a certain chemical that we put in here. What chemical do we put in the auger? I'll give you a hint. It's the first part of the central dogma. DNA, yes. DNA is in there. So we actually put nucleotides in here. So there's a bunch of nucleotides in here, right? So we got our little, double-stranded DNA, what have you. And so we have our bacterial line in here. So we just do a line like this, and we put a bunch of little DNAs in here, and we're looking for the ability to degrade DNA, okay? And if you have the ability to degrade DNA, that means that you will just basically get rid of this DNA around you, but the enzyme will only go so far away from the bacteria because the enzyme only spreads out a little bit, so you'll kind of create this area where you've degraded all of this DNA. The way you test for this, though, is you add a reagent, which is called hydrochloric acid, HCl. And when you put it in here, it actually makes the media cloudy. It causes the DNA to precipitate. But if you have DNase, you're going to degrade all the DNA in here, and you'll create a clear zone. So everything out here will be cloudy, except for this halo area you create, which will be clear. It'll remain clear and not be cloudy because there's no DNA for the hydrochloric acid to precipitate. If you are negative, the entire plate will be cloudy. So if you're positive, you'll have a little halo where you've degraded the DNA and it creates a clear zone. So clear zone is positive. If the entire plate is cloudy, that means you're negative. You do not have the ability to break down DNA. You lack DNase. Does that make sense for that one? I know a lot of people got confused with that, so we want to make sure you guys get that. Does anybody have any questions on that? Yes, no, maybe so? All right. So, okay. Gram-negative test. So I messed up here. I left the old gram-negative tests that we did in here, but I'm going to talk about the gram-negatives real quick. Because this one you don't have to know as much, because the test is a lot more straightforward. So just kind of ignore that, because a lot of it's not relevant for us. If you guys remember, the gram-negative test consists of two types of tests. One is a slant, and the other is that test strip, where we add in a buffered saline solution, and then we tip it forward, and it goes into those wells, and then we just incubate it for a little bit, and then we read the wells, and we add some reagents. What you have to know from this is which wells we add the reagents to, and which reagents. It's the second to last three, so it's like proline, GGT, and PIRE, we add the REML reagent to, and the very last well, we add COVAX reagent to determine if indole is being produced. And that one's the only tricky one, remember, because well 18, it tells you if it's adenylyl positive, so we read that one first, then we add COVAX reagent and read it a second time. On the actual test, if I were to give you guys a test tray, I would tell you, hey, this is what this color is before we added in indole, and then you can have to figure out indole all on your own. Okay. So we read those tests and we figure out which colors they are. The TSI slant is one of the most important ones because it will help you to identify which organism you have by knocking out a bunch really quickly. The first thing it tells you two important things. One, it tells you if it produces hydrogen sulfide or not. There are only 3 hydrogen sulfide organisms: Citrobacter, Proteus vulgaris, Proteus mirabilis. So if you see that black precipitate, that means that there's hydrogen sulfide being produced. You actually know that there's only 3 organisms you have to differentiate. Those 3, Proteus mirabilis, Proteus vulgaris, and Citrobacter. Because those are the only 3 that make hydrogen sulfide. For fermentation, if the slant, if you guys remember the butt part, if that turns yellow, that means you're able to ferment glucose. If the slant part turns yellow, that means you're able to ferment lactose and sucrose. There's only 2 organisms in our list that can only ferment glucose and not lactose or sucrose. That should be Proteus mirabilis and, I believe, Morganella morgani. So those are the only 2 that don't do anything other than glucose. So if you only see a yellow butt, but a red slant, then you know, okay, well, now I've only got 2 organisms to figure it out between. So that's the most important test to kind of chunk what you have in half. If you have the hydrogen sulfide, then it's those 3. If you don't have hydrogen sulfide, you can knock those 3 out. You don't have to look for them anymore because you already know what it is. For the other tests in the test wells, the really important ones are the urease test, and the TET and lipase test. Those are really important. And then the very last important one is kind of like the malonate one. But more or less, you just kind of have to go and get like kind of like the best answer. We will give you test trays that are done very accurately so that you guys will be able to identify them, but be aware, you have to be careful and figure it out on your own. So we will give you guys the little coded paper that has all the colors that tells you what's positive and what's negative for each test. So you'll have that. You'll also have a chart to be able to identify the organisms based off of that result, but you guys have to make the interpretations of what you're seeing into those results, whether positive or negative. The last little bit for that is people tend to get tripped up on that because they panic because there's like a bunch of tests. What I found that people have the most easiest way is to write down in a line, okay, well this one's positive, this one's negative, 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 positive, positive, negative, 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 positive. And then this is like A/A H2S positive. And they just kind of write that down really quickly so that they can kind of go back and figure out what those are. We will have extra stations set up so that at the end of where everybody goes through all their stations, you guys can go back and look at them. We'll have extra stations so you don't like all crowd around the gram-negative identification ones. So you guys can go back and look at them at your leisure. But that's the method that I've seen a lot of students use successfully to kind of like really quickly jot down the answers so that they can go back and think about them in a more clear manner and they're not under a time crunch. All right, does anybody have any questions on this? Makes sense. Okay, how are we doing on time? Not overshooting time by too much, am I? This class is supposed to end at 2:15, yeah? Okay. I don't want to keep you guys extra. I'm sure you guys don't want to be here extra. Okay, bioinformatics. Two major formats. There's two ways we look at DNA sequences on computers. The first one is really quick, also known as FASTA. Very good. Very quick, FASTA. The other one is a little bit easier for us to read as humans on the second half of it, but the first part of it has a whole bunch of kind of like tabulated information that's really hard for humans to read, but very useful with like background information on the gene. That one's got a lot more exhaustive information, and that format is called GenBank. Very good. So GenBank is the one that has kind of like, it's written into columns of 10 nucleotides each, and then each line is numbered. So line 1 goes 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. It keeps going until it gets to 60 and then starts back over again. So it's a really handy format if you need to figure out which position nucleotide 23 is in. FASTA is really easy for you to write manually. In the FASTA format, which you guys should know, is a greater than symbol and then some title, and then a new line, and that new line has your sequence information in it, okay? And it can be any length, it doesn't matter. It's really kind of convenient for that. Okay. What are we talking about? Bacterial identification uses a specific gene. So there's one gene that we use. It's kind of universal to all organisms, especially bacteria, because they all kind of share a very similar organism, similar enzyme. Which enzyme do we use? Which gene are we looking for? It's one of the ribosomal genes. It is the 16S. So it's the 16S subunit for the ribosomes. So ribosomes are very conserved, but they have some regions that have variability, and that variability can help us identify organisms. Not all of them, but a good chunk of them. And we used a universal set of this to make our PCR product. So we used kind of these base things to run our PCR. What is the universal thing that we use? They're called primers. Very good. Primers are short little sequences of DNA, and they stick to the template DNA at kind of lower temperatures than what we would expect for the genomic DNA. So they're very, very handy for that. And we need them to be able to make that reaction occur. The website that we use that hosts the algorithm, what website do we use? It is called a series of letters that's really obnoxious. Do you know what it is? Huh? NCBI. NCBI is owned by the National Library of Medicine, but NCBI hosts the website. So, in case you're going, National Center for Bioinformatics, NCBI, or Biological Information, I think is actually the correct term. Okay, and that hosts the algorithm that we use to search through these databases, and that algorithm is abbreviated BLAST. What does that abbreviation mean? That's an acronym, I should say more specifically. What does that stand for? Does anybody know? Okay, everybody forgets this. It's really important. Basic, local, alignment, sequence, or search, in this case it's search, tool. So everybody replaces the S with like sequence, or they put the T with like something else. Basic, local, alignment, search, tool, BLAST. Make sure you guys know that one. That's important. Hint, hint. Okay. Completely synthetic PCR and gels are referred to as, so things that we do in a computer, we refer to them as what? It's a Latin word, because we have to be obnoxious as scientists. In silico, very good. Which is different than what we normally do, which is like in vitro or in vivo, which are in cell culture or in tissue or in situ if we do it in organism. In silico means in a computer, in silicon. Okay. These websites can make and utilize prediction sequences, and we can cleave these sequences with enzymes. And so what these enzymes do is they basically find specific sequences, and they only cleave that specific sequence. So if we're looking at EcoR1, it cleaves a specific sequence, and it won't cleave it anywhere else. So if your gene product doesn't have that sequence in it, that cleavage will never happen because it won't be there. But if it is there, then it'll make a little cleavage, and now you get two fragments, especially when you run it on a gel. These are very good at predictive stuff, but it's not something we want to rely on too heavily because we have to do real-world actual analysis. When we were using the search algorithm, we used two basic databases. Everybody forgets these because they're kind of not super common. The first one is what? I'll give you guys a hint. It means it doesn't have any copies in it. It is a non-redundant database. So it doesn't have any extra stuff in it. So it's non-redundant. The other one is RefSeq, which is reference sequence. And it's basically saying, hey, this is what we would say is the standard sequence for like all of this organism. So for like humans, everybody has this gene in it and it's exactly sequenced like this, which is of course not true, but we call it, we pretend, we just make believe. We call that a reference sequence. Okay, PCR stands for what? Polymerase chain reaction. Don't call it a PCR reaction like I do all the time because that's dumb. Polymerase chain reaction. Very good. Each reaction contains an enzyme. This enzyme is very, very crucial for making the whole thing work. That enzyme is called what? What is it? Now you guys got quiet. It's a type of polymerase. It is a DNA polymerase, right? So it makes copies of DNA. In this case, it's a specific one called Taq, which is from an organism that we stole it from that lives in high temperature things, Thermophilus aquilus or Thermodactylus aquaticus. Thermus aquaticus is the new name for it. And so we need that really high temperature because we go through cycles to do this. In this reaction, we need short sequences, which we've already talked about. You guys called it out already, which are called primers. So short DNA sequences, and we use it to make a copy of the original DNA, whatever our sample is, and we call that a what kind of DNA? Template. Very good. So that's our template DNA. Often template DNA is our genomic DNA sample. Sometimes it's not. Sometimes it's something else crazy. To do this whole reaction, we use a machine. Instead of sitting there with cups of hot water, we use a machine to do it. That machine is called a what? Thermocycler, very good. Yeah, it goes through cycles of heat, increases and decreases the heat at set time points. In this case, there are three important ones. So it contains three phases. Those three phases are, what's the first phase? Denaturation, very good. The second phase is called annealing, very good. And the last phase is elongation, and it moves through these cycles. It goes through each cycle, and then it goes back to the beginning and does it over and over and over again for 30 or 40 rounds. You should know the temperatures for these cycles. The first one is denaturing, and that's the highest one. We set it up to 95 degrees Celsius so that it pulls apart all the genomic DNA. And the first time we do this is extra long. So the very first one we do is for a very long time. It's about 5 minutes, and that's the only time we do it for 5 minutes. Every time after that, it's really short. It's like 30 seconds. And that's 95 degrees Celsius. So it's really, really hot, melts all the DNA. The second one we do is the annealing temperature. That's about 55 to 65 degrees Celsius, and that lets the primers stick on. The primers can stick on at slightly higher temperatures, so they don't need it to go down that far. And then lastly, we do the elongation or extension phase. Both are fine. And that is where we let the DNA polymerase bind to the primers and the template DNA and start making a copy. And then we go through that cycle over and over again. Okay, let's see here. Last little part. We run this through a gel with electrical current to move the DNA from the negative charge to the positive charge, because DNA is slightly negatively charged. And there's a dye that binds to double-stranded DNA that reacts with UV light or blue light. And I know it's really close to the time that you guys are going to go, so before I end it, does anybody have any questions on anything we got? If you don't, if you still want to come up and talk to me, come and talk to me after class. We'll chat and we'll go over some more things. I'll have probably another little study guide thing popped up, and we'll see you guys next week. And definitely, if you guys have any questions, come and see me.